They come in all different colors, shapes, and sizes.
You can have large and small sponges shaped as vases or baskets or even large barrel sponges. They can be branching, they can be round, they can be encrusting, they can be massive or thin. So the variability of the body plan is pretty amazing.
Many of us have used them to bathe or clean with. And the most famous one is SpongeBob SquarePants, a cartoon character who entertains children from his pineapple in the sea. But have you ever given sponges much thought?
The 500 million years of evolution indicate that they're true survivors.
In recent years, researchers discovered that sponges are one of the groups of marine animals that produce the most bioactive compounds, leading to many medicinal uses. These include the development of drugs that treat viral infections, other infectious diseases, and a variety of cancers.
There's over 8,500 species of sponges that are known. Sponges are found in all ocean basins, from tropical to Arctic. There's a few freshwater sponges, so they are very widespread. Many tropical sponges can be found on shallow reefs to deep reefs. Sponges are animals, primitive and simple. They don't have true specialized or differentiated tissue like a heart or a brain or a liver. They do have differentiated cells. Depending on the type or class of sponge, the skeleton can be made out of silica or calcium carbonate. So most sponges have a silica-based skeleton, and these are in the form of what's called spicules.
These spicules determine the consistency or feel of a sponge.
You can go from very soft to very rock-like. People might learn a little bit from sponges as to how do they survive disturbances, changing climates. Who do you want to go to when you run into trouble? Well, it's a group that's been around for a long period of time, and the sponges fit that bill.
What might sponge genomes tell us about long-term survival? What other secrets might sponges reveal?
Major funding for this program was provided by the Batchelor Foundation, encouraging people to preserve and protect America's underwater resources. And by Divers Direct, Emotion Sports, inspiring the pursuit of adventure and water sports. And by the following, in memory of Harriet Fagan, the Do Unto Others Trust, and the Friends of Changing Seas.
The islands of the Bahamas, a tropical paradise where sandy white beaches lead to crystal clear waters that are home to beautiful reefs and marine life. The rich variety of sponges that thrive on these reefs is of particular interest to Dr. Joe Lopez, a professor at Nova Southeastern University in Dania Beach, Florida.
Sponges are ready for us here. The sponges' role on the reef is variable and complex. They can be food for some predators. Some think they are the glue of reefs because they're in many different parts of the reef. They provide a three-dimensional topography, so they're providing lots of new habitat for other animals.
Sponges don't have a digestive system, Instead, they are filter feeders that sift their food out of the water.
They do this by creating a current through their bodies with specialized cells. These cells have a long tail and it beats, causing water to flow through these specialized canal systems. These cells are able to pick off particulates of food, which in most cases are bacteria, plankton, dissolved organic matter, and that's how they sustain themselves.
To demonstrate this powerful and efficient filtering system, Joe injects harmless fluorescent dye at the base of a sponge.
They're creating this unidirectional flow of water, usually coming in through the sides, and then this canal system eventually empties out into a bigger outgoing opening.
For his research, Joe is collecting tissue samples from various types of sponges. His goal is to compare samples of the same species collected in different areas.
So we take a small section and they can cover that cut and regrow. An important part of sponges are the bacteria and microbes that live within that tissue, because sometimes that forms part of the actual body of certain sponges called high microbial abundance sponges. The microbial cells can make up 50% or more of the whole sponge body, so they're highly dependent on their microbial symbionts.
Joe is curious if these so-called microbiomes of certain sponge species vary by location.
I'm part of a consortium of sponge biologists who are now looking at various species of sponge microbiomes, and this is part of a broader Earth microbiome project, which is very interesting. So we've been able to apply high throughput DNA sequencing to multiple species of sponges, asking various types of questions. Do they vary biogeographically, by host, or by season? It does seem to be that specific sponge species have a specific microbial community. We'd like to see how much environment might affect the composition.
Preliminary analysis revealed distinct differences in the microbiomes of the same species of sponges collected near different islands in the Bahamas. Samples collected off New Providence Island, which is highly developed and home to a relatively large human population, had an approximately 10-fold higher abundance of potentially pathogenic microbial groups than those collected near Andros Island, which is sparsely populated. These initial results show that the proximity of human populations can impact the microbial communities associated with sponges. Joe will also use the samples he's collected for more extensive genomic studies in the future.
Got a couple species, well, one that I've never gotten before. I've preserved specimens for long-term DNA use. So we'll be able to isolate DNA from the host as well as the microbial symbionts. We'll analyze the whole genome. These could be part of the Global Invertebrate Genome Alliance, which I helped found 2 years ago at Nova Southeastern University. This stands for GIGA. The goal of GIGA is to sequence several types of genomes from multiple species of invertebrates, of which the sponges belong. And we'll see a lot of diversity at the genomic level because of different habitats these sponges are found in, the various forms or compounds they produce. I think we'll see some very interesting insights once we read the DNA code of those genomes.
Joe's first trip to the Bahamas was 20 years ago.
I came here as a student in the 90s, and that's what inspired me to go into marine biology. What kind of air they gave us.
Hopefully good hair.
Now he's back for the first time in 10 years, thanks to a partnership with the International Seakeepers Society.
Seakeepers was founded
to get yacht owners directly involved in ocean research and conservation efforts. The most expensive part to doing marine science research is getting out on the water, is being able to be in the field, collect samples, collect data, and get out there long enough to really complete a full project that can result in something significant. And so one of the things that Seakeepers is currently doing is to get more scientists out in the field at no cost or very limited cost to them.
One of the places Joe wanted to return to on his trip is the community of Red Bays, on the northern tip of Andros Island. Andros is the largest, but also most sparsely developed of all the Bahamian islands. Sometimes called the Sleeping Giant, this island is famous for its blue holes, traditional handicrafts, and its sponge industry.
The sponge industry is still alive and kicking in Red Bays. It's a tradition. My forefathers, they used to sponge. My granddaddy, when I was a little boy, he used to always tell me about sponges. Each family has about one guy that goes in the boat sponging.
3 different species of sponges are commercially viable, called hardhead, wool, and grass sponges.
This is a hardhead. This is abundance. You have a lot of abundance, and this is what you call a wool. It's like the price of sponges. This one here, you put about 22 to 23 on a strand, and they get about $6. But this one here, you get about, on a nice solid one, this one is already torn up inside, but you get about $20 for it, if the right buyer comes along.
Is there a certain season?
For sponge?
Yeah.
No, sponge is all season.
All year?
All year for sponge. Leon is your guy.
Leon?
Yeah.
Okay.
He can show you everything, man.
Locals collect sponges on the shallow Great Bahama Bank on the west side of Andros Island.
Well, not many people have been studying the genomics of these bath sponges. So it might be interesting to take a look at how they compare with some other sponges. And maybe we can see how they produce this softer skeleton.
After sponges are cut from the bottom, they're brought back to shore.
So you cut them, take them out, put them on the land, on the sand, preferably the sand, so everything can just drain down through the sand. Sometimes we walk on them, speed up the process, and once all the barnacles and everything get on them, they get light.
After some time on land, the sponges are taken to a crawl in the ocean to soften up again.
The hard edge, you put them on the shore today and you can clean them tomorrow. But the wool and the grass, they take like 7 days on the land, another 7 days in the crawl. And you get in the 7 days, you get in the crawl and you beat them out with your beater and you tap them and squeeze all the mud and everything out of them. And you throw them in the boat and they're good to go for market.
Local spongers sell their product to a wholesaler that ships them to markets all over the world.
Mostly the Greeks that buy sponges.
Sponging was once one of the country's largest fishing industries, until a disease outbreak in the late 1930s wiped out the vast majority of commercially viable sponges, putting many Bahamians out of work.
Some people say it was a disease came about, and it's a bunch of different stories. Some people say the Lord blew his breath on it because certain people were robbing the black man. It's a bunch of different stories. Now they're back again. Been here for a little while, and it just came back to life in the early 60s.
Today's fishery is much smaller, but the local spongers on Andros still take pride in their work and quality product.
This is a beautiful one.
Yeah.
It's a grass.
This
That's
come up
grass?
the grass. This come up the mud. This is
That's a
roll.
mud, that's a roller. It lasts a lifetime. The artificial ones will tear up after you. Sometimes you put some chemicals on or soaps or whatever liquids, and they tear up. But if you get a piece of wool in the kitchen and you're using it on the bathroom, it'll be there like forever.
The only time sponges are able to move around is in their larval stage, when they're ready to hatch from their brooding chambers. But how do these free-swimming larvae know how to orient themselves to find a good place to settle and grow?
Sponges are known to possibly have this protein, which is involved in phototaxis. And that means that it allows the larvae to sense direction and where light is being emitted. Most animals use opsin, another protein, as their photosensing detector and molecule. Now, sponges lack opsin, but they do have cryptochrome. So we're interested in looking in depth into how they possibly use cryptochrome as the light sensor.
The challenge is collecting sponge larvae at just the right time. In July 2015, Joe and his former PhD student, Andia Chavez Fonegra, received word at their lab in Dania Beach, Florida, that sponges on local reefs were brooding, so they set out to collect samples.
It was great.
What's good, what's cold?
Lots of sponges.
They have the brooding chambers, they have the larvae. It was successful, little chambers of larvae inside of the sponges, so that's good. These are the brooding chambers. The white things that we are seeing there are the eggs, and some of them are fertilized, and they probably will be in process of becoming a larvae, and that is what we want.
So this is a great find. It's the first time we've found larvae. They'll have this black spot, and this black spot could help them phototaxis towards the light. So it's a kind of eye, and in this case it's a non-neural eye. There's no neurons or advanced developed cells that would be involved in vision.
Experts want to know how this fundamentally different sensory system functions. To find out, Joe has teamed up with collaborators from the University of California.
We're trying to keep some larvae live, and we're going to ship those out. Others we're going to keep for RNA analysis and DNA analysis.
Together, they plan to conduct genetic, physiological, and behavioral tests to determine if cryptochrome is indeed the light receptor in these sponges.
We're going to see if this molecule called cryptochrome is being expressed actively. But this is the first time we've looked at it in this species, and we can connect that to the physiological studies if it's active and expressed.
Understanding the function of this non-neural eye will provide fundamental new insights into sensory biology. Until recently, there was scientific consensus that sponges were the first animals to branch off the animal tree of life.
Darwin first wrote in The Origin of the Species that there is a tree that connects all living organisms, and people have been trying to put together this tree for years. So the tree of life is trying to show where all these organisms are placed. So they're grouped according to similarities. So you have reptiles distinct from mammals. Sponges are generally placed at the base of the animal part of the tree that then lead to more complex animals.
The key thing is that everything is connected, and at the tips you have animals that live today, and then there's branches that don't really make it to the tips, and those would be extinct animals.
Dr. Joe Ryan is an assistant professor at the Whitney Lab for Marine Bioscience in St. Augustine, Florida, where he studies evolutionary genomics.
Traditionally, animals were grouped by their morphological characteristics. So animals like primates, who have relatively large brains to their body size. That would be used as a character to unite primates. Sponges don't have very many characteristics that are similar to other animals. So basically all other animals are thought to be more closely related to each other based on that morphological evidence. And later with microscopy, cell types were used to kind of fine-tune it. At the base of the tree, there was a single-celled ancestor to all animals. And of course, going from single cells to a multicellular organism is an increase in complexity. So the next step would be an animal that has multiple cells but doesn't have a nervous system, doesn't have a musculature system, so is more of a passive type animal. And sponges, their morphology and their cellular composition all fit well with this idea of simple to complex progression in animal evolution.
As technology advanced, scientists also started using DNA sequencing to help build the tree.
We're all connected by DNA. Now, if we can read the sequence from all these organisms, we can theoretically put together a tree of life of species that are existing now. And we do that by counting the similarities between the genes and what they show.
This has led to a surprising discovery that has some experts casting doubt on the theory that sponges were the first animals to branch off the tree. In 2008, a group of international scientists conducted genetic research suggesting that ctenophores, also known as comb jellies, are an older lineage.
So it was a big discovery. So ctenophores are animals that live in salt water.
Despite the fact that ctenophores look like jellyfish, the two are not related.
There's about 200 described species, many more that are known that haven't been described yet. And they are found from the North Pole to the South Pole in deep water, shallow water.
The initial research was received with skepticism by many in the scientific community who felt more data was needed.
I was one of those people who was in that camp. Once there's more data, this maybe will change back the way it was. But what happened is that the more data you throw at the question, the stronger the result is.
Joe Ryan and his collaborators decided to expand on the initial research findings.
We sequenced the genome of the warty comb jelly to say, okay, what happens when we rerun these analyses but with a complete set of data? And the position of ctenophores as the sister group to the rest of animals held. And then we also started asking, well, what if we look at different ways of looking at this data? Do we get a different answer? And that also came out in favor of the ctenophores as the sister group. So we had independent evidence and we confirmed some existing ideas.
But if ctenophores did indeed branch off the animal tree of life first, several long-held theories about the evolution of animal traits are thrown into disarray. Historically, it was thought that simpler animals like sponges, which do not have muscles or a nervous system, were the building blocks for more complex animals higher up the tree. Ctenophores, however, do have a nervous system and muscles, so if they came first, how does that explain the lack of these features in sponges? Scientists have come up with two hypotheses. Either ctenophores developed a nervous system independently from the rest of the animals, or sponges lost theirs.
Either of these are really difficult to understand because we think our nervous system is the greatest thing in the world, right? So if the ancestor had it, then how was it lost in sponges? How could an animal lose a nervous system? And also, it's incredibly complex, the nervous system. How could it independently happen twice?
There is precedent for organs evolving more than once. Eyes, for example, are known to have evolved several times in different species. But there's also a precedent for animals losing seemingly vital organs.
There are cnidarian worms that are related to jellyfish, and they've lost their nervous system. Perhaps sponges started off as these filter feeders that live on the bottom of the ocean, and having a nervous system was a high cost from a metabolism point of view. If you're trading off between filtering and sensing your environment with neural cells, it becomes better to invest more energy into the filtering.
Further research is needed to settle the debate over who branched off first from the animal tree of life.
There's so much data out there with this advanced sequencing that it's going to take time to clearly make sense of it. But I think the final word is not out. I mean, I think we still need to look at all the data together. I believe in total data sets. Now, the hard thing about working with these very old lineages is this is very deep time, 600 million years. So it's tough.
I think it's important in science to explore all the possibilities. But, you know, I'm open to all the possibilities, right? You have to be.
Major funding for this program was provided by the Batchelor Foundation, encouraging people to preserve and protect America's underwater resources. And by Divers Direct, Emotion Sports, inspiring the pursuit of adventure and water sports. And by the following, in memory of Harriet Fagan, the Do Unto Others Trust, and the Friends of Changing Seas.